Okay, fantastic. We'll move to the second talk by Tomas Knapen, Mapping the Dark Side. Very catchy title, Thomas. Mapping the Dark Side, Visual Selectivity of Default Network Deactivations.
It takes a while, but oh, there we are.
Okay. The dark side,
So I want to thank the organizers for allowing me to speak about this and organizing a really cool conference in general. So what I'm going to talk about is some findings of ours that we as visual scientists find very interesting and weird, so I hope you find the same thing. I'm a vision scientist. Most of us are in, I think, some sort of sensory type of interest in sensory regions of the brain. So I think talking about the default mode network, it needs some explanation. So what is that default mode network? It's a network of co-activating regions around cortex that are that normally find in resting state experiments. And if you do like a task-based experiment, you would generally find that it activates during things like mind wandering and autobiographical memory. Now, there's studies showing that if you have people listen to a story, semantic categories that are represented in these regions are things like social concepts, again highlighting that the sort of computations that happen in these regions are very high level and abstract. Now, furthermore, if you look at just the spatial layout of the cortex, you find that exactly these regions in the default mode network are furthest removed in terms of geodesic distance from the primary areas of cortex, both sensory and motor. And that puts them in a position to do a lot of multimodal integration, for example. Now, I've seen the default mode network appear in my own experiments, and I'm a vision scientist. So what happens if you do a vision science experiment, and for example, you have people report an unexpected change in a visual stimulus? You see that not only do you get activations in frontal and parietal regions that are associated with attentional processing, you also see these very consistent deactivations in the default network, and that's that dark side, what that dark side refers to. Now, usually people don't look at this because, negative stats, you don't look at it in fMRI, but there's some signs that these deactivations are functional. They seem to play a role in mnemonics, like in memory processing. From the pattern of these deactivations, people have shown that you can decode whether people are going to remember the memorandum. But if you go to sensory processing, to perceptual processing, there are some intriguing indications that these deactivations play a role in that type of thing too. So for example, in this experiment, the deactivations seem to correlate with the amount of surprise about that perceptual occurrence. And also, you're able to decode whether people will perceive a low contrast stimulus or not from these same regions here in the angular gyrus. So given that there are these indications that these deactivations play some sort of functional role in perceiving visual stimuli, we wanted to ask the question whether we can use sort of standard vision science tools that we know and love to understand the functional organization of these default mode network regions. So what I'm going to show you is an experiment in which we do population receptive field mapping. Just to show you the broad outline of what that sort of experiment entails, here I'm showing on the left a visual stimulus, or I will be showing a visual stimulus on the left, and on the right you see a flattened representation of cortex. So if we play it, we will show structure visual input, which in our case is a bar traversing the visual field, and you see traveling waves of activation going through primarily visual cortex and frontal and parietal lobe. Now I want to get into the task that we had our subjects perform a bit, because it's important for the logic of the later analyses. What people did during the experiment was they fixated. While they fixated, the bar traversed the visual field. On every TR, this bar is shown at a specific location, and in the middle of the TR, all the gabors that compose the bar are given a color. And we can change the ratio of red and green to blue and yellow elements inside the bar, which we can titrate. So the task that the subject performs is a color discrimination task, and they do this once every second, or a little less than a second. So they're continuously engaged in this task directed at the stimulus, and we run these runs. We have blank periods in which people are just steadily fixating, and these bars traverse the screen in different directions. It's important to say also that we titrated the task difficulty to be the same across the visual field. So we have separate staircases titrating task difficulty for different eccentricities of the bar position. And that allows us to say that whenever there is a visual stimulus on the screen, people are equally engaged with the task. Right? So in terms of task engagement and stimulus presence in general, you could think of this as a very simple block design fMRI experiment. I want to invite you to just fixate the center of the screen here, and just to show you what the experiment is like. You try to answer the question red, green, blue, yellow for yourself, and you notice that your attention is being drawn along with that visual stimulus. And so this is what people are doing for about 1.5 hours at a time. So what's really nice about this type of experiment is because you have such structured visual input, you can sort of zoom in and analyze each separate voxel independently. And what you see is what you measure is these voxel time courses. You see that whenever there's a stimulus on the screen, you get these activations. This is a voxel from area MT. And you see that whenever there's a stimulus on the screen, you see these activations. What we can do is try to fit the exact spatial location that this voxel likes by assuming that its response is due to a population receptive field, in this case shaped as a two-dimensional Gaussian. We can change the parameters of this Gaussian, like the location and the size of it, and then fit the time course. Right? So for every voxel, what we get is a set of parameters. And those parameters we can visualize then on the surface of the cortex, for example, to show, in this case, polar angle. So you can convert the polar angle of its visual field location to a color, and you see these nice polar angle progressions on the surface of the cortex, allowing you to delineate visual field maps. Okay, so that's our technique, but what does it have to do with the default mode network? If you show a visual stimulus like this, and keeping in mind that the task was similarly difficult or equally difficult whenever there was a stimulus on the screen, you would say, okay, these default mode network activations, if they represent task engagement, you would expect them to cause signals in an on-off manner. So you can analyze a voxel time course as a block design. So what I'm showing you here is a voxel time course from angular gyrus. And you see that if I fit it with a block design, you get a negative beta weight. So it stands for a deactivation of this voxel's response whenever there's a stimulus on the screen. It's also very clear that there are signal deviations that are not explained by this non-spatially specific model. So what we can do is apply our trick, right? We try to fit a spatial receptive field, but in this case, we allow its amplitude to become negative during the fitting procedure. So it's a very simple change to our standard procedure in which we fit retinotopic precise voxel responses. So if you do that, you find that you can much better estimate the, or much better get towards the precise time courses of these voxels, meaning the signal is much better explained by assuming that it's caused by a spatially specific deactivation instead of a spatially general deactivation. What that means is that these voxels seem to report visual field location by means of their deactivations. Now, I can do this for every voxel, of course. What I'm plotting here is just the sine of the amplitude of the PRF multiplied by its cross-validated R². And for the remainder of the talk, I'll only be presenting voxels in which the cross-validated R² of the spatially specific PRF model is higher than that of the on-off model. Now, if you look at these flat maps, for example, you see that they're not really well, they're nice, but they're not really precise. And if we want to look exactly at the parameters that we're fitting from these regions, we're going to have problems. For example, if you're going to look at the angular gyrus region, there you definitely see veins running through the results. So this was scanned at 7 Tesla, and we see a lot of these veins running through these regions on the surface. It makes it relatively hard to really say whether there are map-like structures in the data. So what I'm going to do is segue to a separate data set that was recently put online. It's the HCP RETINOTOPY data set, put on bioRxiv, and the data are downloadable, which we did. And it's a really, really beautiful resource. So it's 181 subjects that performed retinotopic mapping, on which retinotopic mapping was performed, and 10 minutes of those are more or less the same experiment that we performed, apart from the attention task. So this is exactly the same type of plot that I'm showing you here as in the previous slide, but now it's based on the average of 181 subjects. You definitely see, again, I'll be zooming into the angular gyrus, that there are negative spatially specific responses there, and I can convert those, or then show those polar angles in a similar way as I did before for the visual field maps. And I'll just zoom into those quickly. What you see is that these negative responses are spatially specific. Not only spatially specific, they seem to encode visual field locations that are contralateral to each hemisphere. Now, this sort of visual intuition we can quantify by means of drawing polar angle histograms, which I'm doing here for positive rep maps in the interparietal sulcus. Right, so these representations are polar angles colored separately for each hemisphere. You see that the left hemisphere represents contralateral visual space, and vice versa for the right. You can then calculate a contralaterality index, which is near 1. So this is for positive retinotopic maps. This is the type of thing that you would expect for a standard retinotopic map. If you do the same for the angular gyrus, you see more or less exactly the same pattern of polar angle distributions. Now, another property of visual field maps is that whenever a cortical location, a neuron or a voxel in this case, responds to a visual field location close to the fovea, it will have a smaller receptive field. And whenever it's further in the periphery, it will have a larger receptive field. Now, this translates into a very nice correlation for standard visual field maps between the size of a population receptive field and its eccentricity. Right? So plotting here eccentricity on the x-axis and size on the y. This is for IPS on the left and what we might say is frontal eye field on the right. We can do the same analysis again for the angular gyrus region, and we find very similar eccentricity size correlations. So these are two sort of principles that hold for general visual field maps, and they seem to also hold for these default network regions. I want to now segue back into our own data set and then perform a decoding analysis. If these localized deactivational responses are functional, then as a region of interest, a field map should represent visual space. And by performing this decoding analysis, we can actually verify whether that's the case. So we're going to do some cross-validated leave one out analysis where we fit PRFs on all but one run. And then we combine from a test set, a left out run, per TR, the bold pattern from a given region of interest with these fitted PRFs and estimate the most probable stimulus given that pattern of bold responses. And we're using a version of a method developed by Van Bergen in the orientation domain, which uses both the overlap between the PRFs in the spatial domain, that's the feature space covariance there, and the structure of the residuals after the fit. Now, this method allows us to reconstruct the visual stimulus from brain activity, and I'm showing you an example from area V2 here. So you really get movies for the entire experiment where you see this bar passing by on the imagined screen. You can do this for each region. This is V2. It has smaller receptive fields, so the bar looks really nice. The larger the receptive fields in a given region, the more vague this will naturally be. Now, in this analysis, if you do that, again, I'm showing IPS, so positive retinotopic maps on the left. What you get is you can take the peak of that reconstructed stimulus and plot it in X and Y coordinates. And early, so here's when the bar was moving from the bottom to the top. Early is colored purple, and yellow is for a late stimulus presentation. So you see that there's a different spatial pattern there. You can collapse this across one axis. You're talking about screen coordinates here, so you just rotate for every bar pass. And then what's nice is that these decoding analysis just amounts to a little, like a simple correlation between actual bar position on the screen and decoded bar position from a given region of interest, but with a 4.5 second HRF delay. Now again, I can do the same analysis for the angular gyrus region, showing exactly the same type of pattern, and a very strong correlation between actual and decoded bar position. I can do this for all regions in the frontal and parietal attention network on the left, and for 3 sort of standard regions in the default mode network. And we can decode where a visual stimulus is from each of these regions. So to conclude, what I think this may, or what I think we show is that these default mode network activations that people see in many, many experiments, that they're retinotopically specific, meaning that they report visual field location of a given stimulus. And you can conceptualize that as them being negative PRFs. And as an aggregate across a region of interest, they do seem to represent visual space. Now, what does that mean for what happens in the brain when you show a visual stimulus? What I think it means is that when you show a stimulus, it activates in a spatially specific way the visual system, of course. But what this means is that it also deactivates in a spatially specific manner the default mode network, right? And what that sets up is this plus-minus thing that we know from many types of sensory coding, and it allows us to think of this in terms of a contrast that, in terms of computations, might function to decorrelate things. Now, if you think of it as a sort of contrast like this, the sign of that contrast might determine the balance between cognition being directed externally towards the sensorium or internally towards things like memory, for example, or social concepts. Now, what I think is most interesting myself is that it harks back to this very old saying going back to Aristotle, where we can say nothing is in the intellect that was not first in the senses. And specifically, what I think this may mean is that the brain repurposes sensory reference frames to sustain higher level cognition, and perhaps this would be a way to investigate higher level cognition using these reference frames. So I want to thank my PhD student and postdoc who are instrumental in this work, and the funding agency, the Dutch funding agency. Thank you.
Questions? Otherwise, I'll just leave you off to the coffee break, and you can harass Thomas over a coffee if you want.